Hello, this is Donna Mayer, Executive Director of the Cholangiocarcinoma Foundation. I'd like to welcome you today to the second in a series of webinars about our new Research Fellowship Program, which was established in 2015 to support early career researchers who focus on studies in cholangiocarcinoma. Our goal is to raise awareness about cholangiocarcinoma and inspire innovative quality research. If you have a specific question for the presenters, you can enter it in the box on the right of your screen, and then I will ask it on your behalf. The presenters will not be able to answer any personal questions today during the presentation, so please be sure your questions are general in nature. Our first presenter is Dr. Allyson Merrell, the recipient of a $50,000 grant named in memory of Andrea Scott, who was a young mother of two. Allyson received her bachelor's degree in physiology and developmental biology from Brigham Young University. In 2014, she received a PhD in human genetics from the University of Utah. She is currently a postdoctoral fellow at the University of Pennsylvania in Dr. Ben Stanger's lab, studying the reprogramming of liver cells and the formation of cholangiocarcinoma. Welcome, Dr. Merrell. I now turn the floor over to you.
Thank you, Donna. Thank you for the opportunity to talk about my work today. So today I'm going to tell you about my work to determine the cellular origins of cholangiocarcinoma. Cholangiocarcinoma is a cancer of the bile ducts, and it's classified as either intrahepatic, perihilar, or distal cholangiocarcinoma based on its location. Today, for the purposes of this talk, I'm only going to be focusing on intrahepatic cholangiocarcinoma. The American Cancer Society estimates that 2,000 to 3,000 people will develop cholangiocarcinoma each year in the United States. Most of what's known about the cholangiocarcinoma cell of origin, however, is based on the physical appearance and gene expression of the tumors, not on molecular lineage data. So the purpose of my project is to expand our understanding of the cholangiocarcinoma cell of origin, or the cell type in which it develops, in order to hopefully improve patient outcomes. We hope that a clear understanding of the cell of origin may allow earlier detection, possibly of these tumors, and improve predictions of tumor behavior. And ultimately, we hope that this will lead to preventative therapies. So we hope that as we better understand the different cell types that lead to cholangiocarcinoma, we can better target our research and treatment options in order to better develop patient outcomes. Before I talk more about the cell of origin, though, I first want to tell you some more about the liver. The main function of the liver that most people are familiar with is detoxification of chemical compounds. However, the liver also plays another essential role in the production of bile for digestion. So in the liver, bile is produced and then transported through bile ducts, shown in this image in green, to the small intestine to help with digestion. And this bile is produced by the hepatocytes, shown here in tan, that are the predominant cell type of the liver. These hepatocytes will produce bile salts and other bile components that will ultimately travel through the bile ducts, shown here in blue, which are lined by biliary epithelial cells. And these two cell types, although they have very different functions, actually originate from the same developmental source. During development, both the hepatocytes and the biliaries originate as hepatoblasts, or fetal liver cells. And these hepatoblasts that are next to the portal vein will receive signals to become biliary epithelial cells. These hepatoblasts that are next to the portal vein will eventually form a tube, including the lumen that the bile duct, or the bile, will be able to travel through. The rest of the hepatocytes that are unable to that don't become biliary cells will develop into hepatocytes. Previously, it's been proposed that these two cell types, the hepatocytes and the biliary cells, will each give rise to a different liver type, a liver cancer type. So by appearance and gene expression, hepatocytes most resemble hepatocellular carcinoma, suggesting that originally that this particular cell type, the hepatocyte, gave rise to hepatocellular carcinoma. Similarly, intrahepatic cholangiocarcinoma appears to look like bile ducts, suggesting that this tumor may arise from biliary cells. And so it's been difficult from previous studies which have modeled this in mouse that to understand how this is developing, because they would knock this out developmentally in the hepatoblast, making it impossible to tell whether these tumors are arising from the hepatocytes or the biliary cells. So what you would expect if this model is correct and biliary cells are forming intrahepatic cholangiocarcinoma, if you were to target these genetic mutations specifically to the biliary cells, you would expect that they might form cholangiocarcinoma. And in fact, that's what a study from Stuart Ford's lab found, that when they deleted P53, a tumor suppressor, specifically in the biliary cells, and administered a toxin, a carcinogenic toxin to the mice, these mice developed intrahepatic cholangiocarcinoma. Furthermore, they used a yellow fluorescent protein, or YFP, marker to label the biliary cells so that they could track them and see if they gave rise to the cells of the tumor. And you can see that in this image here, in which green represents the YFP positive cells that have given rise to the tumor. So this shows that given the right genetic conditions, biliary cells can form cholangiocarcinomas. Surprisingly, though, two studies have also found that hepatocytes are able to give rise to cholangiocarcinoma. So a recent study from Atsushi Suzuki's lab found that when they genetically labeled either the hepatocytes or the biliary cells and administered a toxin to mice, these mice developed cholangiocarcinoma. What they found, interestingly, though, was that only labeled hepatocytes would give rise to tumor cells, and they never saw biliary labeled cells giving rise to the cholangiocarcinoma. This suggests first that hepatocytes are capable of giving rise to cholangiocarcinoma, but also that the particular genetic background may influence whether or not biliary cells are able to produce cholangiocarcinoma. Interestingly, though, these two studies have used different markers to label the biliary cells, which suggests that it may be important to do studies side by side to control for that kind of variation. In further support of hepatocytes as a cell of origin for cholangiocarcinoma, another study from the Willenbrink lab found that both activation of Notch and AKT in the hepatocytes could produce cholangiocarcinoma. And you could see this when they labeled cells, again with the yellow fluorescent protein, although in this case it's shown in red, that these cells express a marker, CK8, of cholangiocarcinoma tumors. So this shows that both hepatocytes and biliary cells have been shown in different situations to give rise to cholangiocarcinoma. So the first aim of our study is to determine if there are conditions in which only particular cell types can induce cholangiocarcinoma, and in which cell types these particular mutations may or may not induce cholangiocarcinoma. So we'll test in different combinations hepatocytes or biliary cells in order to determine if they form cholangiocarcinoma. In order to do this, we need to use genetic tools to specifically label and mutate either the hepatocytes or the biliary cells. So using these specific genetic tools, we can fluorescently label the hepatocytes, shown here in green, but avoid labeling the biliary cells, shown in pink. Alternatively, we can specifically label biliary cells and induce any genetic mutations, while none of the hepatocytes will be mutated or be labeled. So using these genetic tools, we can test different combinations of mutations in order to determine whether the cellular origin of cholangiocarcinoma depends on a particular genetic background. So we'll first activate AKT signaling and Notch in hepatocytes or biliary cells and determine if these can develop tumors. It's known from previous studies that AKT and Notch in hepatocytes can form cholangiocarcinoma, but it's not clear whether or not under these conditions biliary cells may also form cholangiocarcinoma. Next, we will activate KRAS and P53 deletions in order to determine if this can also give rise to cholangiocarcinoma. These are genes that are very frequently mutated in human cases of cholangiocarcinoma, and it's been shown that developmental activation and deletion of these in hepatoblasts in mice can induce cholangiocarcinoma. Because this was done developmentally, however, it's unclear whether this activation and deletion of these genes induce tumors through the hepatocytes or the biliary cells. And so we will test these to see which condition can give rise to cholangiocarcinoma, or whether both may. Lastly, we will also test the role of liver injury in the formation of cholangiocarcinoma cell of origin. So we will administer a toxin to mice, as well as delete the tumor suppressor P53, and determine whether cholangiocarcinoma in any tumors arise from hepatocytes or biliary cells. So what we would look for in all of these cases will be whether these genetic mutations are able to give rise to cholangiocarcinoma. And what you would expect if the particular genetic background is critical, that certain conditions may produce cholangiocarcinomas, while others require a certain cell type that only hepatocytes or only biliary cells are able to give rise to tumors in those conditions. We are currently in the process of breeding mice to be able to conduct all of these tests. Now, I'm going to tell you it gets actually a little bit more complicated than when one cell type gives rise to a tumor, because in fact, hepatocytes can actually change their cell type when they're stressed by injury or signals. So recent studies from our lab and the lab of Marcus Grantee at OHSU have found that hepatocytes can become biliary cells following injury or signaling changes, such as ectopic activation of Notch signaling. And during this process of reprogramming, the hepatocytes turn off genes specific to the hepatocytes, such as HNF4 alpha, and actually begin to turn on biliary genes, such as SOX9 and ultimately CK19 or cytokeratin 19. And so you can actually look at this happening in vivo using genetic labeling. So in this image here on the right, all of the hepatocytes have been labeled in red. And what you can see is that following this activation of Notch signaling, some of these red hepatocytes have become biliary cells and have turned on the biliary marker CK19. So for AIM-2, what we want to focus on is whether cholangiocarcinoma forming hepatocytes first reprogram into biliary cells. So cholangiocarcinomas formed from hepatocytes take on the appearance of bile ducts, suggesting that these hepatocytes must be changing their fate a little bit. Furthermore, many of the stresses that cause reprogramming are also known to induce cholangiocarcinoma, such as Notch signaling or injury. And so in AIM-2, we want to determine if reprogramming is a characteristic of hepatocytes that are forming cholangiocarcinoma. And so our hypothesis is that as hepatocytes become malignant and turn into cancer, that if they happen to receive signals to undergo reprogramming, are more likely to become cholangiocarcinoma, whereas hepatocytes that don't receive these reprogramming signals would become hepatocellular carcinoma. So in order to test this, we'll take advantage of a mouse model in which AKT activation within the hepatocytes has been shown to specifically cause hepatocellular carcinoma. Similarly, in another mouse model, we would use that has been shown that Notch activation and AKT activation as well induces specifically cholangiocarcinoma when mutated in the hepatocytes. So by comparing these mice, we can look at the two different tumors, hepatocellular carcinoma and cholangiocarcinoma, and determine if markers of reprogramming are present in these tissues. So what we would expect is that if reprogramming is a hallmark of cholangiocarcinoma formation, we would start to see hepatocyte genes turning off and biliary genes turning on in these tumor cells. And what we would expect if hepatocellular carcinoma forms from an absence of reprogramming, that we wouldn't see any of these markers in this cell type. And so in AIM-2, we hope to determine if there's evidence of reprogramming being a critical step in the formation of cholangiocarcinoma. So in conclusion of what I've told you today, both biliary cells and hepatocytes can give rise to cholangiocarcinoma with particular genetic mutations. But a thorough testing of different conditions is necessary to determine if the different cell types only form tumors with particular mutations. Also, I've told you that hepatocytes can undergo reprogramming into biliary cells following injury. And many of these same injuries and signaling changes can also induce cholangiocarcinoma. And this suggests that hepatocyte reprogramming may be a hallmark of hepatocyte-derived cholangiocarcinoma. And we hope that a better understanding of cholangiocarcinoma and its cell of origin can help to improve patient therapies. That understanding which cell type produces tumors with particular mutations may allow us to better predict tumor behavior and target therapies to cancers with different origins. Also, if reprogramming is a critical feature of cholangiocarcinoma development from hepatocytes, we can target therapies to interrupt this process under pathological conditions. And with that, I'd like to thank the Stanger Lab, in particular Ben Stanger, for allowing me to work on this project, and the rest of the lab for their support. And I'd especially like to thank the Cholangiocarcinoma Foundation for funding my research and allowing me to pursue this project. Thank you.
Thank you, Dr. Merrell. The first question that we have is, do you see the KRAS being a predictive test in the future?
Certainly. I think it's something that has been a test in a lot of different cancers. I know it's very critical in the formation of pancreatic cancer as well. So I think that could definitely be a great marker to use for treatment in the future.
Okay, thank you. And then the next question is, do you see, or is the treatment for hepatocellular cancer the same as for cholangiocarcinoma?
I believe they're similar, although I don't have a lot of experience with that. But from what I understand, the survival rates are much higher with hepatocellular carcinoma, and that it's a little bit possibly easier to treat.
Okay, well, that's all the questions that we have for you at this time. I'm going to introduce our next presenter, who is Dr. Chad Walesky. His proposal won the Innovation Award, which was funded for $60,000. Chad is a research fellow at Harvard Medical School, Brigham and Women's Hospital in Boston, Massachusetts. He is currently working on understanding signals that tell cells within the liver to become normal liver cells or bile duct cells. His group is hoping that understanding these signals will allow for not only a better understanding of cholangiocarcinoma formation, but also new targets for drug development. Welcome, Dr. Walesky. I now turn the floor over to you.
All right. Thank you, Donna, for that introduction, and thank you all for listening. I also want to thank the Cholangiocarcinoma Foundation for not only their support, but for giving us this great opportunity to talk about our work. So, like Donna mentioned, I'm a research fellow within the lab of Wolfram Gosling at Brigham and Women's Hospital and Harvard Medical School. So this project title is a mouthful. So really, what this distills down to is we're trying to attempt to talk to cells and talk them out of becoming bile duct cancers. So what I mean by that is we're trying to understand different signals within the liver that are telling normal liver cells to be normal liver cells, or those signals that are telling cells to become bile duct cells. And then in understanding all those signals, can we at all try to talk cells out of becoming bile duct cancers? So, as we know, cholangiocarcinoma is a cancer that affects the liver. And I'm here to tell you the liver is a really cool organ, and it has all these really great functions. I've worked with the liver my entire career so far. And so the liver is an essential organ. We need it to live. I guess that's why they snuck the word live in there when they were naming it. But the liver has all these amazing functions. One that we learn about usually in middle school or high school is that the liver creates this stuff called bile, which helps us in digestion, right? And in particular, digesting fats. So one of the functions of the liver is to produce that bile. Another function that the liver has is that it helps to break down and store many vitamins and chemicals that are essential for the body. Now, one of these that I've got listed here as an example is glucose. So it's actually essential for kind of maintaining the normal glucose levels within our blood. Glucose is also known as sugar, and our cells need that sugar to make energy. So another function that the liver has is it makes substances that are required for the body to function normally. One example of this is blood clotting factors. So the liver makes these blood clotting factors that then float around in our blood, and when we get a cut, they become activated, and that helps stop us from bleeding. And then another feature that the liver has is that it helps clean up our blood. And one of the most common associated things with this is alcohol. So the liver helps to clean up, all the alcohol or medications or drugs that we intake. So as I mentioned, the liver makes the stuff called bile. And the bile is made within the liver cells out here. And then it gets transported to the structure called the gallbladder, where it's stored and then secreted for when we need it. So like if we eat a meal, especially one that's high in fats, our bile will get secreted and then will help us with the digestion of those fats. But with all of that said, we need kind of this plumbing system, or this system of tubes or pipes to help get that bile that's being made, even all the way out here at the periphery of the liver, the way back into our digestive system, and that's where the biliary tree comes in. It functions as this kind of plumbing system to collect that bile to transport it to our digestive tract. So cholangiocarcinoma, as we know, is a cancer of those bile ducts. And as Allyson mentioned, there are about 2,000 to 3,000 patients in the U.S. each year that get diagnosed with bile duct cancers. Now, it's considered a rare cancer, but 2,000 to 3,000 people a year is pretty significant. Bile duct cancers have a 5-year survival rate as low as 2%, but this really depends on the type and location of the tumor, and this can be much greater. And as was mentioned previously, there are 3 major types of bile duct cancers. Intrahepatic bile duct cancers, which are bile duct cancers, they're really associated with these smaller bile ducts within the liver. Perihilar bile duct cancers and distal bile duct cancers. So I have intrahepatic bile duct cancers highlighted here because we feel that right now what our story is telling us is that it may be most associated with intrahepatic bile duct cancers, but we're going to investigate more in the future to see if it applies to these other types of bile duct cancers. So right now the treatment options for cholangiocarcinoma are listed here. We can usually use surgery to try to resect the tumor or to remove it. This can be combined or used with a radiation therapy. So we use radiation to try to shrink the tumor or kill the cells. Also, we use chemotherapy, which is the use of anti-cancer drugs to try to stop those cells from growing or to kill those cells. And also there is best supportive care therapies where we may not be using curative therapies to try to eliminate the tumor, but we're trying to really add to the quality of life of patients. And this can actually extend life expectancy as well. And this can be something like inserting drainage for bile ducts. But really what all this highlights is that we need a better understanding of cholangiocarcinoma to develop better treatment strategies. So within our lab, we use the zebrafish as a model system and as a model organism. So the zebrafish has many advantages as a model, especially of cancer. So this right here is a picture of 2 zebrafish. They're named zebrafish due to their really pretty stripes that they have that are zebra-like. So this is a male and a female zebrafish. The male here has this more golden color. The female has a more pale white color. And you can see the female also has a little bit of a distended abdomen where she's carrying the eggs. So this pair of zebrafish can actually produce as many as 200 offspring in a week. So that gives us an enormous amount of organisms to actually study. Also, zebrafish are genetically very similar to humans. They also produce transparent embryos that develop outside the body, so we can actually watch them as they develop. We can watch organs as they grow and as they develop. And also we can treat these zebrafish with chemicals just by putting them in the water. So we can treat the embryos with different chemicals to see how it affects liver development and bile duct development. And also we can treat adult zebrafish to see, if a certain chemical may inhibit the growth of tumor types such as cholangiocarcinoma. So what this diagram over here on the right is showing is that many of the same organs that we have, the zebrafish has. And in particular, we have a liver here in the human. We also have a liver here in the zebrafish. We also have a heart, a kidney, spleen, intestine. So they have there are a lot of similarities. So the zebrafish liver actually forms, they actually form a functional liver by 3 days of age. And it's very similar to the human liver. It contains all of the same cell types, such as normal liver cells and bile duct cells. So this is actually a picture of a zebrafish embryo. This is the same embryo on the left that it is on the right. And what we're using here is a technique called fluorescent microscopy, where we actually can shine certain special types of light on the embryos. And these embryos are special in that they are producing a molecule within their liver cells that when we shine that light on them, their liver cells will show up this really pretty red color. And then they have a special molecule that's being expressed in their bile duct cells, so that when we shine this special light on them, the bile ducts show up this green color. So we can observe the development of their liver cells, their bile duct cells, all in real time. So what this slide is highlighting is that we can actually develop cholangiocarcinomas in zebrafish. So we can generate these cholangiocarcinomas and study the signals that contribute to their formation. And we can also test chemicals, like I mentioned earlier, to see if they can at all inhibit cholangiocarcinoma formation. So these pictures here, you're seeing the tumor with the zebrafish has this distended abdomen here, and whenever we dissect back the skin, you can see the liver is really enormous. It's blown up with a tumor, and whenever we look at this under a microscope, we can confirm that this is cholangiocarcinoma that's forming. So you get all of these nice kind of tubular structures, these bile ducts that are overgrowing, those bile duct cells that are growing, confirming to us that this is cholangiocarcinoma. So I mentioned earlier that we use the zebrafish a lot for developmental studies. And oftentimes, developmental signals can shed light on pathways that may also be altered during cancer development. And it's easy to assume that cholangiocarcinoma may fit this model because the liver cells and bile duct cells both develop from a common precursor cell. So what do I mean by that? So here, this is an illustration of our common precursor cell, which can then become either a bile duct cell or our normal liver cell. Now, one thing that we think might be happening is that during cholangiocarcinoma formation, we get kind of a push toward developing into bile ducts versus our normal liver cells. So what we're trying to understand are the various signals that are important for telling this common precursor cell to become a bile duct, or the signals that are essential for telling our precursor cell to become a liver cell. So in trying to understand this, we've actually shown that we can influence the outcome of these precursor cells and whether or not they'll become a normal liver cell or a bile duct cell. So what this is again our red liver cells under that fluorescent microscopy and our green bile duct cells forming here, both outlined with these yellow ovals. And when we give a certain drug, we can actually push the livers to develop only into the green bile duct cells. So here we lose those red liver cells, and we maintain and can make an argument that we actually increase the number of green bile duct cells. So really, when we look back at this illustration, we're seeing that we're beginning to understand the different signals that are pushing our precursor cell to develop into our bile duct cells preferentially. So what this data is showing us is that we're actually seeing the opposite, because it would be really cool if we could, push the development of those precursor cells to our normal liver cells versus the bile duct cells, because in cholangiocarcinoma, we want to start, shrinking tumors and losing the bile duct cells. So this again, is our red liver cell under fluorescent microscopy, our green bile duct cells. And this is a drug that actually functions much in the opposite manner of the one that I previously showed. So we get, this is a gradient of increasing drug amounts from left to right. This is our normal red liver cells. They're maintained with increasing amounts of this drug, and we can actually lose the expression of these green bile duct cells. So we're pushing these livers to develop primarily into our normal red liver cells. And this is illustrated here, showing that we're understanding more about these signals that are causing our precursor cells to be pushed to our normal liver cells. So ideally, we want to understand how all of these signals work so that we can get back to a normal balance, right, of our bile duct cells to our liver cells. So what's next? So what we want to understand next is this mechanism at all relevant to cholangiocarcinoma development? And our goal is to understand this relationship in more detail, and in great detail, to understand signals that can create a normal balance of liver cells and bile duct cells in hopes that we can develop new drugs and have new drug targets. So with all of that said, I want to thank everyone that was involved with this work, my mentor, Dr. Wolfram Gosling, and everyone within the Gosling lab, and also our collaborators, and of course the Cholangiocarcinoma Foundation for their support and this opportunity. So I'll thank everyone, and I'll take any questions.
Thank you, Dr. Walesky. There's one question, and it's just in regards to where and if genetics plays a role in your study.
Yeah, so what we're trying to do is trying to actually understand how some of these, so you can align certain mutations, genetic mutations, with the drugs that we're actually using to treat these zebrafish, and we're actually showing this within genetic backgrounds too. It's kind of hard to explain, but yes, there's very much a genetic component to this that we are exploring, and I hope that answers your question.
Okay, yes, thank you. That's all the questions. On behalf of the Cholangiocarcinoma Foundation, I'd like to thank both Dr. Merrell and Dr. Walesky for sharing their time and expertise. We will be posting this recorded webinar and PowerPoint presentations on the Cholangiocarcinoma Foundation website. I want to thank all who participated today. The webinar will end now.